Okay, it's really great to see a full house, but not at all surprising. We want to hear, Antónia Monteiro, hails from the sunny land of Portugal, and I actually remember hearing her talk a long time ago when I was a PhD student. We heard there's this amazing professor coming, in the next door department, and we all went to hear this, so literally and metaphorically beautiful research done with, butterfly wing patterns, and that was Antónia. Just when you moved to Yale University from Buffalo, I think, yes, and since then you moved to Singapore when Yale started this Yale-NUS. Is it a liberal arts college we together established? Yeah, that was such an interesting sort of development. They ended, this year. Oh God. But then, Antónia, you also had the joint appointment with the Department of Biology, and where she is a professor, endowed professor now. And so she's here as a visiting scholar. Just arrived not long ago, a few weeks ago or so, and then you'll be here until New Year. Yeah, is it right? Yeah, correct. And you are up for meetings to discuss all the interesting, exciting ideas. So you're available for, coffee, lunch, and any meetings without any eating. And so please catch her and take this opportunity in the next couple of months. It's wonderful to have you here, and so the floor is yours, Antónia. Go for it.
Thank you, Naomi. I want to start by thanking the theoretical visiting program and the people that allowed me to come here for a sabbatical. So I'm here on sabbatical. I'm primarily writing a book on the evolution and development of butterfly patterns, but I'm also here because I've started working on behavior, larval behavior, odor-mediated behavior, looking at how behavior changes patterns of gene expression in the brain. And Okinawa and OIST in particular has an incredible concentration of neurobiologists. And so I've been gradually sending emails to different people, trying to meet them over lunch. And I will continue doing that, but I will also be very happy to talk to anybody who is interested in what I'll be talking about today, which is my bread and butter, where most of my grants come from, which is still about the evolution and development of wing patterns in butterflies. Okay, so I'm presenting you here an image. Let's see if I can figure out the pointer. Does this work? This pointer, maybe not. This does not either. So I think I'll just use my, it might not be interpreted correctly. Okay. So I'm showing you an image of a Junonia butterfly. And one of the things I want to get to in this talk is to show you the diversity of mechanisms that exist to pattern the wing of a butterfly, but then how these patterns can be quite distinct as you go from the front wing to the hind wings, and then also quite distinct when you turn the butterfly around and then you have completely different wing patterns on that other surface. So I think we've all seen butterflies in our milieu. They have incredible colors. Some of them are pigmentary colors, others are structural colors. And then they use these colors to produce these incredible complex patterns on their wings. And we might start by asking this more basic ecological question: why does wing pattern diversity evolve in the first place? And the general answer will be, okay, butterflies live in a variety of different environments with different light qualities, different backgrounds, meadows with different colors, and so forth. So they're colonizing our planet in different habitats. But there's two primary goals that a butterfly has. And one of them is to survive until the adult stage, to find a mate and to lay eggs. So there's two imperatives that the butterflies need to fulfill before they die. So they have to graduate from the caterpillar state into an adult, and they eat as fast as possible, develop without being eaten by anybody. And then they have to use their wing patterns to find a mate and to avoid being predated as adults as well. So these are the two imperatives of the life of a butterfly. And the butterflies have decided to fulfill these imperatives in different ways. So they've invented different strategies to survive and to find mates. So one of the strategies is exemplified here by this Delia's butterfly, is just to use bright, vivid colors as warning colors, just like the mushroom is a warning, bright red, don't eat me. So a student in my lab, she did this paper model type of experiment, very simple, where she takes the butterfly and prints it, the original butterfly on paper. But then in Photoshop, she removes all these bright colors and transforms them into gray values. And then one at a time, just the yellow becomes a gray value, or the red becomes a gray value, or removes all the black and the bright colors, and then puts them in the forests in Singapore and tests which of these paper models that are attached to a live bait, a little mealworm, get eaten first, and which survive the longest. And she finds things like, if you are the wild type pattern, you survive the longest. But if you retain the red, that's the next longest time that you survive. And then the yellow is the second longest. And then all the other patterns don't give you a lot of benefit. So the mealworms get plucked off from these paper models faster. But then there's also amazing strategies like this one, masquerading. So here's a moth in Malaysia that was sitting just outside of our hotel door. This is not our photo, but we found a specimen of this moth, and it's just astonishing. It has kind of a fly drawn on its wings. And then on the hindwings, there are this sort of splotched colors that look like bird poop. So here we have a moth that's signaling, here's a fly feeding on bird poop. This is not a profitable item to eat because this is poop. There's a fly eating it, so don't come close, don't eat me. So this is what we call masquerading, a different strategy altogether. And then, of course, there's crypsis, trying to look like a dead leaf to avoid being predated by visual hunting bird predators, for instance. And then other wing patterns, the ones I study the most, are these eye spot patterns that are sometimes used for deflection. And I'll just show you a quick video to show how this works with real predators predating on butterflies. So this is a mantid that is going to attack a bicyclist butterfly. This is a wet season form of the butterfly. This is the same species, but the dry season form. And here's a mantis attacking the dry season form. So it scooped up the whole butterfly and started eating the body. But here's the wet season form with more prominent eye spots. And the attack was directed at the wing margins where the eye spots are. And the butterfly was able to fly away with alive. So this was a more slow version. And these are the quantitative data for eye spots. So most of the attacks that mantids do on these butterflies go for the hindwings where the eye spots are located. A few others go for the forewings in red, where the eye spots are also located. And then a very small proportion of the attacks actually target the body of the butterfly. Now, eye spots have this function of deflecting attacks of predators away from the main body parts and into the wing margin. But we asked whether they could also be intimidating. In this case, intimidating means warning predators or telling predators not to even try to attack. So to preventing any attack whatsoever. And Sebastian did these experiments also with paper models in the field, where you wrap a little bit of a wire around a paper model. Again, you load it with a live bait. If you don't put the live bait, these experiments don't work. Nobody notices anything. And then the idea here was to use paper versions of the real species that exist in Singapore, and then remove some of those eye spots, vary the number of eye spots, but most importantly, change the size of these eye spots in some of these more extreme paper models. And what we found here, when in each place of the forest, we placed a single one of these alternative models together with a model that did not have any eye spots, just a cross white band. What you see here is that eye spots, when they're small and many, or a single one, these models get predated more often. So the mealworm gets extracted more often than the paper model without any eye spots at all. So having eye spots might not be always a good thing. It attracts attention from predators and the mealworm gets eaten. But as soon as the eye spot becomes this size, then these paper models become much less attacked than paper models without any eye spot at all. So size also matters. Now, to finish off this introduction about wing patterns in general and eye spots in particular, I'll just show you an experiment where we also identified that eye spots are also important for male-female sexual signaling. So they are important for the two sexes to sort of find each other and decide to mate with each other. So let's see if I can get this to go.
It's ultraviolet flashing as a way of selecting their mates.
Okay, I'll remove so this was an experiment where I'm picking a Bicyclus aninana, an African butterfly, separate their wings, and then I'm using a plant compound, rutin, that I can dab on top of the eye spots and remove their ultraviolet reflection. So these white centers that a lot of Satyrid butterflies have under this UV camera, they're extremely bright. And this white UV brightness seems to be what males and females are picking on when they are deciding to mate with another individual. So if you block them and you put two males in the cage with a female, one of the males has the UV signals blocked, the other male doesn't. The female very, very often will mate with the male without the blocked UV signal. So eye spots, especially on the dorsal surfaces of the wings, seem to be involved in sexual signaling. And dorsal surfaces of wings, which are the wings that are hidden for the most part when the butterfly is at rest, these surfaces seem on average to be the ones that are used for sexual communication. So these morpho butterflies that everybody loves from South America, Costa Rica, etc., they have this incredible distinct pattern. So they have these beautiful iridescent blue colors on the hidden surfaces, and then they use their eye spots on the exposed ventral surfaces that are constantly exposed to visually hunting predators. So there is this incredible diversity of patterns that appear in different sides of the wing. So the dorsal surfaces are private surfaces for male-female communication, because when the butterflies court, when males court females, or sometimes females court males, they come very close together, and often the courtship movements involve rapid flickers of the wing and displaying the interior surfaces of a butterfly wing. So those are where the more sort of striking colors often evolve. So one of the things that I want to do in this general talk is just guide you through the evo-devo mechanics for how is it that butterflies can actually evolve different wing patterns, different colors on different surfaces of their wings. So here's an example. Each butterfly is split down the middle. And what I'm placing here is just the ventral side on one side and the dorsal side of the same species next to it. So you can see that dorsal ventral patterns are one of the main axes of variation in terms of color patterns in all these different species here. There's another axis of variation. Oh, before I go there, the other axis of variation that is already visible in this slide is that front wings and hind wings also often have differences between them. So in this case, you can see the butterfly in the middle there on the top, orange front wing, and then striped blotched hind wing, completely different. So both these two axes, dorsal, ventral, forewing, and hind wing, are exemplified here. And then here again, but here we have males and female axes. So we have males on this side, and this would be what the female wing would look like. So butterflies have been evolving across all these different axes and sexes. And what I'll be telling you about today is how do they do this? So how are these patterns first controlled within the wing? How do you start by coloring different parts of the wing in different ways? How do you start subdividing a wing into different regions for applying these colors? And how then is this mechanism altered as you go from one surface to another or to a different sex? I'll give you a little bit of detailed on how a wing develops. So wings actually start developing within the body of caterpillars. So the wings are developing inside segments, thoracic segments 2 and 3. They're very tiny in the up to the final instar, which is what this 5th instar is. But at the beginning of the 5th instar, you can already take these wings out and you can start examining some of the patterning processes that are happening. And then in the phase called the prepupal stage, the butterflies, the larvae stop feeding. They hang themselves by the abdominal end here, and the wings grow exponentially inside their body until when the animal pupates, the wings are now visible, and they're this big surface area here, directly accessible to experimentation to the outside. So they are on the surface of the animal, and they're very large pupal wings. And you can see here, for instance, there's these little bumps on the wing cuticle that indicate where the future eye spots, for instance, of that species are going to be developing. So if we take the very tiny wings in the beginning of a 5th instar larva, here I'm staining for 2 different genes, and these are HCR stains. So what we're looking at are mRNA messenger molecules attached to fluorochromes. So it's a hybridization chain reaction type of protocol. And what you can see in the green gene, which is called rhomboid, it's a gene that is expressed in trachea. You can see that the trachea start invading a very simple wing blade that's just 2 layers of epidermis, and the trachea are tubes that invade that 2-layer epidermal epithelium and start branching into more and more branches, subdividing the wing into these different, what we call wing sectors. So nothing like this seems to happen in Drosophila. So this is a very distinct process of what's happening in Drosophila. This process of trachea invasion into the wing pad of Drosophila is very late in development compared to what's happening in butterfly wings. And then this gene called blistered seems to be expressed in red gradually in between these epithelial cells that are being subdivided by the branching and growing trachea into these little lobes. So this process essentially is creating these divisions of cells that are going to be vein-like cells or intervein areas. And there's no difference at this stage between different parts of the wing. But as the wing gets bigger, you start to see the expression of genes that are continue to be bound to those regions where rhomboid penetrated as a special group of cells that migrate and penetrate tissues, just like trachea penetrate the whole body of an insect as the insect is growing. So there's going to be a series of genes that continue to be restricted to those vein areas. And then there's another set of genes that are expressed also from the very beginning at the edges, at the wing margin. And so genes like wingless in pink here, or vestigial in blue, and another type of wing signal. So these signals, just like Drosophila, are super important for making the wing grow out. So these are important signaling centers of the wing for outgrowth of the wing. So this is happening, the subdivision of the wing into these marginal gene expression patterns and more internal patterns is happening at the same time that these patterns of gene expression dividing vein and intervein tissue are happening. And then on top of that, there are signaling centers like DPP, which is a morphogen, the same morphogen that is visible in Drosophila wings and one of the earliest patterning genes in the Drosophila wing. You can see it also in the butterfly. There's actually 2 sources of DPP, not a single one like in textbooks of development in Drosophila. And then this DPP morphogen is probably setting up a series of slices of gene expression, OMB in green, Spalt in red, that subdivide the wing into different type of colors and flavors along the anterior and the posterior axis. And as this is happening, eye spots, and I'll show you that in a minute, are also being differentiated as all of this is happening in a wing. So the process of eye spot differentiation, we've been modeling it using reaction diffusion equations. And the way we do it is we use a signaling group of cells of like wingless signaling cells in the wing margin, represented here by this orange color. And we put boundary conditions within each of one of these wing sectors bordered by veins. And if we have a kind of DPP, another morphogen, in this case represented in blue, if we have DPP present everywhere in the field of cells where wingless is signaling from along the margin, and specific boundary conditions on the vein areas, we can have a stabilization of a third gene called distillus, starting with marginal expression, rising up into a finger, and then stabilizing in the middle between 2 veins in the middle of each of these compartments. So distillus, as you can see there in orange, is that orange gene that seems to be expressed potentially through this reaction diffusion mechanism, specifying cells in the middle of nowhere in between these two veins. Now, when you differentiate these cells in the middle of nowhere by expressing distillus, and then many other genes seem to follow, that's just the specification of a future eye spot center. These cells become incredibly important signaling cells. So if you transplant them, for instance, in early pupation, if you move those cells around and you plop them somewhere else on the wing, they are sufficient to induce rings of color around them, induce an eye spot around them. So what happens between this larval stage, when the center is differentiated, and the adult, when you have all these rings of color around the eye spot, is that there's this intermediate step where we see a bunch of different transcription factors being expressed in the pupal stages of development, when the wing is much bigger and at the surface of the animal. We see these Spalt in this case in the red here, and then optics, a gene that is involved in eye development as well, expressed in the surrounding cells. These same genes also expressed at the center banding pattern of the wing, and so forth. So there is potentially, we still don't have a good handle on how this differentiation step is done. The model that most people follow in this field is that these cells at the center expressing distillus and other things might be expressing DPP and wingless. One or both of those morphogens might activate these transcription factors in a concentration-dependent manner, setting up these rings of expression. And then 7 days later, after this stage, which happens right after pupation, within 1 day, finally, the pigments are going to be synthesized in these cells. But these cells differentiate into their particular domains very early into the pupal stage. And then you end up with, during development, these cells that are marked, their nuclei are marked with these colors. These cells, each of them creates a projection, a cuticular projection that leaves the epithelium and creates this thing called a scale. And it's inside the scale that these pigments are going to be deposited. And this is the powder that you get on your fingers when you touch a butterfly wing. So each scale is the product of a single epithelial cell. And the nucleus of the cell is left behind in the epidermis. But this projection is the thing that's on the outside surface of the wing, and presumably evolved, we don't know, but maybe when butterflies hit spider webs, they kind of have this, they don't stick. There's a powder of scales that gets attached to the silk, but the butterflies bounce off the spider web and continue flying without their wing scales. So this may be another adaptation that is specific to this group of insects. Scales can come in many different shapes and sizes. Some of them have incredible nanomorphology. I'm not going to show you that here, but they can produce these iridescent blues through ways of bending light. So they are not pigmentary colors, they are structural colors. Okay, so back to the main topic. I've shown you.
Can I ask a question before?
Sure.
I was curious about this part about development. How much this is specific to one butterfly species and how much is it conserved across butterflies?
Yeah, so there's different groups working on butterfly wing patterning. These patterns that I've shown here are probably going to be quite conserved across different species. But for instance, this pattern here, this band in the center of the wing only is present in some species, but not others. although it's called the central symmetry system in the butterfly nomenclature. A lot of species have it. It's called the system of bands in the center of the wing. It's the central symmetry system. This is called the border symmetry system, and only nymphalid butterflies have eye spots. So this is a novel trait that evolved only in the nymphalid lineage of butterflies.
When the pattern is the same, do we get a sense that there are the same genes? It's caused by the same genes.
Yes, yes. Yeah, so the wing margin genes are well studied across a variety of different species. They're always there. Wingless is always there. DPP is always there, things like that. So these are pretty general patterns. Okay, now why do different wing surfaces display different wing patterns? Now I need to orient you again to the quadrants of a butterfly body. And so if I extend my arms, this is my forewings, my legs would be my hindwings. And so these are important things here, the front wings, the hind wings. And then if I turn my body, this is my ventral surface. If I turn my body, this will be my dorsal surface. And we ourselves, just like butterflies, we have different things happening on the ventral and dorsal surfaces of our appendages. So we don't have nails growing on the ventral sides of our fingers. The nails only grow on the dorsal sides of our fingers. So we have that type of distinction. And interestingly, the genes that are involved in dorsal ventral differences in butterfly wings is the same gene that is involved in dorsal ventral differentiation of our limbs, of our own hands and arms. So Apterus A, it's the name of this gene in butterflies. It's only expressed on dorsal surfaces of the wing. So if you cut the wing and you put it embedded in wax and you stain for this gene Apterus, you see it in epithelial cells on the dorsal side, but not on the ventral side of the wing. It's a transcription factor. It codes for this Apterus A protein. And if you knock it out with CRISPR-Cas9, here's an example of what we did with our own species of African satyrid butterfly. The wild type dorsal pattern looks like this. The wild type ventral pattern looks like this, with silver scales over there and a bunch of eye spots on the hind wing. When you remove a gene that is only present on this surface, Apterus A, with CRISPR, with Cas9 and guide RNAs, this surface starts to develop eye spots everywhere. The forewing starts to develop ventral specific silver scales, and the whole thing starts to become like the ventral side. So it's a super important gene that creates asymmetries on the wing, on the dorsal ventral side of the wing. And when you remove it, you essentially have the same genetic program taking place on the two epithelial surfaces of the wing exactly the same way. So it's one of the most important asymmetric proteins in development that a lot of different gene regulatory networks, for instance, the gene regulatory network involved in building eye spots, it's a gene that has made connections with these networks to create all kinds of asymmetries in wing pattern between dorsal and ventral surfaces. So again, Apterus A seems to be a repressor of the genetic program to make silver scales, which look like this under SEM. They don't have any holes in them. They have two clean surfaces of cuticle. The brown scales, typical ones, have all these windows, all these holes. When you look down on them from the top, silver scales don't have that. And this gene prevents silver scales from developing in that area. As soon as we remove the gene, the silver scales appear. So we call it a silver scale repressor. It represses the ability of these individual cells to produce this morphology, and without it, then they produce this, this brown scale morphology. And it's also an eye spot repressor, because most of the time, there are very few eye spots on the dorsal surface. When you remove the gene, eye spots appear. So it's a repressor of the eye spot gene network. Another interesting gene is ultrabithorax. So ultrabithorax is the Hox gene. We have an equivalent gene in our own bodies. We use these Hox genes to pattern our ventral column to produce different somites along our vertebrate column. UBX is one of these Hox genes that is present in the T3 thoracic segment 3 of insects, and it's homogeneously expressed on hindwings, but is not present at all in front wings. And again, when we do CRISPR targeting this gene, and I have to point out that these CRISPR experiments produce mosaic wings, so not all the cells in the wing get mutated for this gene, only subsets of cells get mutated. And it's through investigating many of these mutants that we infer what the complete action of this gene does onto the wing. And what we inferred was that it's a gene that is also involved in repressing silver scale development. So if the gene is removed, silver scales start developing in hindwings just like they do normally in forewings. And it's also seems to be a promoter in this case. UBX promotes eye spots on hindwings. So normally when UBX is there, and this is a wing where UBX is functioning normally, this eye spot is happily there. But when you remove UBX in this clone of cells here, the eye spot is gone. So in this case, UBX functions as a gene that promotes eye spot development in hindwings, especially in these sectors. And if you see a forewing, it does not have eye spots in those sectors, and it does not have UBX expressed in it. So UBX is a gene that also contributes to make wing pattern differences between forewings and hindwings. So in this case, we discovered that UBX is a silver scale genetic program, a repressor, and it's in this case a promoter of eye spot development on hindwings where it lives. Now, this promotion of UBX might be somewhat indirect, because we discovered this interesting gene called TBOX6. This gene is a gene that we also have, we have TBOX5 and TBOX4 expressed in our front limbs and TBOX4 expressed in our hind limbs. This is a similar gene in the bodies of butterflies. You can see the pink color, it's expressed very highly in the sectors on the forewing that will not develop eye spots. But this pink gene seems to be faded in the sectors of the hindwing that will develop eye spots. So one possibility here is that this UBX promoting effect on these eye spots is through this gene. So perhaps UBX represses the pink gene over here, and then by repressing the pink gene, it allows eye spots to appear, whereas no such repression happens on that front wing, and so the pink gene is there and is repressing potentially eye spots on those two sectors of the wing. Okay, the final topic. Differences between males and females. How are those achieved at the genetic level? So butterflies have an interesting gene called double sex. Double sex in our own bodies, the closest gene to double sex is DMRT1. It's an important gene for sperm development in males. Females do not, and for testes development in males. Females do not have DMRT1 in humans, and so it's an asymmetric protein in our bodies. Males have this protein, females do not have this protein. This protein is downstream of the sex determination pathway in humans that starts off with SRY being expressed in the Y chromosome, which is only male specific. So it's a gene that's downstream of SRY. In butterflies, that gene is called double sex. In butterflies, both forms, both males and females have double sex protein, but butterflies splice this protein differently. So males have one version of the protein, females have a different version, a longer version of the protein. So the splice exons of the male protein are the green rectangles, and the female includes, so the bottom bit, the three green bits are below are the male version. The female includes 2 extra exons when making this double sex protein. So it's a bigger, more complex protein with a different shape. Now, this protein is what butterflies primarily use to regulate their wing patterns differently in male bodies and female bodies. This is some differences that we find on the wings again of this African satyrid butterfly. I'm pointing to silver scales again on top. Those are only found in male wings. Female wings don't have those silver scales. Males, on the other hand, have these very interesting, very long scales, hair-like scales, that they use to spread pheromones during courtship. So they flap their wings, and glands that produce pheromones are quite close by to these specific hair tufts, and they help, the tufts help to spread the pheromone over the female butterfly. So females don't have those hair pencils, nor do they have the glands, the pheromone glands. So what happens when we knock out double sex? So here what we're doing is we're going to knock out double sex using that guide RNA, using CRISPR again. So we're going to knock out the protein and an exon that is used by both of the sexes. So we are essentially creating a knockout protein for both sexes. So they are not going to have the male version. The males are not going to have that version. The females are not going to have this version either. So we're eliminating this and we're eliminating that in one go. So when we eliminate the gene altogether, what we find is that the silver scales revert back to brown. So they become female-like, the butterflies, without the double sex protein being there in these cells. And what's surprising to us is that the females become male-like. So the females start producing these hair pencils that typically are found only in the bodies of males. So what did this tell us? It told us again that the male version of double sex is important to promote silver scale development, and only males, because only males have that protein version, can activate silver scales in their wings. And when we don't have it, we don't have silver scales. But it also told us that double sex at all, this protein, has nothing to do with the development of these special types of scales. Because you remove it, the scales are still there, And they appear, I mean, what it tells us is that the female version of the double sex protein, so the female version of the double sex protein, is an important repressor of the scale program in the bodies of females. When we remove that female version of the protein, then these default hair scales appear in the wings of females. Okay. So, and nothing happens, by the way, when you remove it, double sex in the wings of males. So they retain their hair pencils as well. So here's the function summarized for these two different versions of the protein. Double sex, the male form of double sex promotes silver scales in males, and the female version of that protein represses hair pencils in females, creating the sexual dimorphism that we see between males and females. All these proteins, Apterus A, UBX, double sex, they are regulatory proteins. So their function is to bind regulatory sequences, flanking genes that build other things in the cell. And the idea here, just to sort of wrap it all together, is that butterfly wings have one wing patterning program. It's one program, but then they use these three key proteins to tinker with that one program to create these asymmetries between dorsal, ventral, forewing, hindwing, and male and female programs to create more specific things in each of these surfaces. So for instance, imagine you start off early in evolution where with one butterfly species, ancestral to all the other butterfly species, and it has this one wing program. And it's playing its role in all four wings of the butterfly, the forewings, the hindwings, the ventral surface, and the dorsal surface. But then this Apterus protein comes along, and it's expressed primarily in dorsal surfaces. And over the course of evolution, let's say Apterus starts binding regulatory regions, for instance, of this vein-dependent gene, this purple gene. And so starts interacting with this regulation of this purple gene. And maybe this interaction is a negative interaction. So over the course of evolution, you might lose the expression of the purple gene on the dorsal surface, but keep it intact on the ventral surface. And then again, UBX, it's a protein that lives only on the hindwings, both dorsal and ventral sides. Over the course of evolution, if you wanted to do something different between vein-dependent patterns in your hindwings versus your forewings, you could evolve binding sites for UBX in regulatory regions of this purple protein. And by connecting the two, UBX connecting to this purple protein in its regulatory way, it might, for instance, repress it, but it would do it only in the hindwing and would not affect the purple expression in the forewing. And again, the same thinking for binding sites evolving for just the male version of double sex, allowing the purple protein to disappear just in male wings, but be retained in female wings. And you can imagine that over the course of evolution, these types of interactions between these three key differentiating selector genes and one wing patterning program evolved in many different ways in different lineages of butterflies to give them unique and distinct wing patterns by regulating a myriad of different genes involved in different processes of patterning a wing, and probably gradually over time creating and accumulating differences in each of these lineages. So in summary, butterfly wing patterns display this amazing complexity shaped by both natural and sexual selection. This complexity is orchestrated by important signaling centers, cells in the margin, along the veins, and so forth, potentially use of multiple reaction diffusion mechanisms to set up different secondary signaling sources of morphogens and so forth. And then these three key genes with surface-specific, wing-specific, or sex-specific expression motifs modify this basic program to alter wing patterns in specific ways. And these are my funding sources, the people that do a lot of the work, all of the work in the lab. Many of them are in this photo, and I'll take any questions. Thank you.
Thank you so much for amazing talk and questions, but please wait for microphone because there is a Zoom. Right, hands up, right.
Thank you for the great talk. It's really relevant to my work, and I can see, well, I'm citing some of these examples. And my question is about the co-evolution between different eye spots in the same butterfly. So is the strength of correlation between evolution of different eye spots stronger within wings or within the same side of the same wing, or is there similar correlated evolution between wings or between sides? Do we have any of such macroevolutionary analysis yet?
That's a great question. So in my early life as a PhD student, I spent a lot of time doing artificial selection on eye spot size. I picked one eye spot on the wing, and I selected up in a high line, what I call the high line, selected downwards in the low line. And what I observed is that the genetic variants that I was selecting on just that one eye spot were influencing the size of all eye spots on the wing, on the same surface, on the opposite surface, and also on the hind wing. So my understanding is that because there is one shared gene network, all eye spots are also using it, the same shared gene network. But of course, as you've observed, not all eye spots are the same size. So over the course of evolution, eye spots have also been, have been modified potentially by these different selector genes. How did I come out of this entirely? There a way I could go back? Sure, I can go back. So over the course of evolution, even though the eye spot gene network is a repeated serial trait, because there are these sector-specific genes that cover only certain sectors of the wing and not others, interaction between those genes and the eye spot gene network might have led to some eye spots being smaller in particular sectors, bigger in other sectors. And so there you can break the correlation because you have asymmetries in the background genetics of a wing. And those genes can interact with the eye spot gene network just in those sectors. And so you can break the correlations apart. But on average, my experience of selecting on one eye spot is that everybody changes in concert. They are all part of the same network. And most of the genetic variants that are embedded in that network affect all eye spots at the same time.
Thank you very much. I'm looking forward to more discussions later.
Okay, looking forward to it.
In one of your earlier experiments, you showed that the wings with the large eye spots didn't get eaten as much as the ones with the small eye
Yeah.
spots. And then later on, you showed lots of pictures of butterflies with small eye spots. So why don't they evolve just one huge eye spot?
Good question. I think it's all about the ecology and, and different sets of predator guilds in different places where these butterflies live. So I think one huge eye spot might be deterrent to some predators, but they might not be deterrent at all to birds or lizards. But if you are living in a forest with lots of little mantids that are small insect predators, they seem to, I mean, it might be better to just deflect the attacks to these small little things that really resonate with the mantid's brain. The mantid brain really thinks, okay, this is where I need to attack. There's nowhere else to go. This is where I need to attack. If they had like one big thing, I wonder if the mantid would be that deterred. I don't know. We haven't actually done big eye spots with mantids, but we do know that when we did experiments with lizards and birds, they attack both wet season forms, dry season forms in exactly the same way. They just gulp them up. These predators are not the ones that shaped the evolution of the many small pattern. It's invertebrate predators for sure, or something else. And different predator guilds will select for different optimal patterns. So we just need to know what the predators are, which we don't really know yet very well. Yeah.
Hello. Just a naive question about caterpillars. They also have eye spots, right? Is, again, there any correlation between those caterpillars with eye spots and the adults? And then what functions, and are you interested in the development of the eye spots in caterpillars?
I have a student right now dissecting the caterpillar eye spots and doing a whole transcriptome study to see, and it's in a species that has both larval skin eye spots and adult wing eye spots. So he's dissecting both the wing eye spot area, the caterpillar skin area, and many other bits of the body to see how they cluster together later, to see if they actually cluster with each other and if they're using the same genes, the two very different types of eye spots. For now, I cannot answer that question.
So it's
yet.
not known if there's just like a correlation between.
We don't know yet. No,
Thanks.
we don't know.
Final question, maybe, or and then maybe on Zoom. Is there any question on Zoom? Is there any question on Zoom?
No.
Thank you very much for the talk. So if I understood correctly, there's some seasonal variations.
There is, yeah.
So do you know, have you looked, any commentary on the mechanism for variation there?
Yeah, so we've looked a lot into the previous people from where I did my PhD. Paul Bregfield's lab primarily did a lot of experiments in the field to show that this phenotypic plasticity was adaptive, meaning they reared both cohorts of butterflies, the ones with the large eye spots, the one with the small eye spots, simultaneously. They marked the butterflies with numbers, they released them into African forests, and they did mark recapture experiments to show that in the wet season form survives the longest, and the opposite in the dry season. In terms of the mechanisms for how they shrink during development, it's primarily controlled by temperature. So temperature in the around the wandering stage, when the caterpillars are starting to wander away from food, looking for a place to pupate, at that moment, when we do temperature shift experiments and we move individuals for 2 days to a lower temperature or a higher temperature in that window of development, that regulates the size of the final adult eye spot. And what happens at that time is that there is a peak of ecdyson that is different in height between the two seasonal forms. And if we inject ecdyson into the form that's going to form small eye spots, the eye spots get bigger. So there is a window of opportunity there for ecdyson to regulate the size of the eye spot central cells and later on the size of the rings as well.
Thank you.
Yeah.
Other questions?
It was interesting that different features of the eye spot were under selection, same for predators and for sexual reproduction. And I was wondering if those two on the eye spot, if those two ever come into conflict with one another, they demand different things from the eye spot, or is that not a problem at all?
Yeah, I mean, it's an interesting question. We have been manipulating eye spots everywhere on the wing in this one species that we rear in the lab. And we did 13 different experiments where we eliminated eye spots from every surface in turn. And the ones that we found that were involved in sexual signaling were just the hidden ones on the hidden surfaces of the wings. Those eye spots, I think, are secondarily evolved. So when we have reconstructed the ancestral wing patterns of nymphalids with eye spots, the earliest eye spots on the wings of butterflies were in 4 or 5 on the ventral exposed hindwings. That's where eye spots originally appeared on nymphallid butterfly wings. So a big phylogeny with 400 species of nymphalids. We photographed them all. We reconstructed ancestral states. 4 or 5 eye spots on the hindwing. That was the original position and display, which leads me to think that that original function was just for predator avoidance. And secondarily, in this lineage of soterid African butterflies, the eye spots were able to penetrate that apterous A barrier, which is a repressive gene on dorsal surfaces. It was able to somehow open holes, evolution, open holes of apterous A expression and allowed eye spots to appear on those dorsal surfaces and now have this novel function of sexual signaling, of being used to communicate with the opposite sex. Whether the original 4 or 5 eye spots on the hindwing had both functions that then were partitioned to different wing surfaces, we don't know. So we need to find those species that still have that pattern today and test whether they use their eye spots both for predator interactions and sexual signaling. We haven't done that yet, so we cannot answer precisely your question. Yeah.
Right. So I'm sure you have a lot of, many other interesting questions too, but, Antonia's going to be around for next 4 months.
Yes, so I'm sitting on the 5th floor, EF floor of the next building, S5, L5.
Yes.
Please come in and visit or send me an email.
Find her, email her, and,
I'm happy to do lunches with everybody, discuss things over lunch, yeah.
And so, Antonia, welcome to OIST.
Thank you.
Great to have you here. And please join me in thanking Antonia for this.